Hello, I'm A1 teacher, SAT2 Biology teacher, Topcue Lee, and I'm going to cover part 1 of cells today. So first thing, we need to learn about the discovery of cells. On the SAT2 Biology, they do ask history questions. On the left, we have Anton von Leeuwenhoek. He invented the first microscope, and he's the first person to discover cells, but he couldn't determine whether they were living things or not. Next, we have Robert Hooke. He also helped in the discovery of cells, and what he did was he named cells. He named them after cells in a monastery where monks lived because they looked essentially the same as what cells look like. So the first cell theory was established by Virchow, Schleiden, and Schwann. And what they stated was all living things are made of cells. So anything you can think of, whether it's animals, plants, tiny bacteria, they're all made up of one or more cells. And then the second is cells are made from other cells. So we can't just create cells out of nowhere. What they do is they undergo a process of mitosis and meiosis to produce more cells. And these we'll learn about in a later lecture. Okay, so when we're looking at cells, they're very tiny. You would imagine, okay, if the cell can do a lot of different things, why can't we just be made up of one cell? Well, the main thing is the surface area to volume ratio has to be right. Because if, as you see in the picture below, if we have a tiny cell, we're assuming that a cell is like a cube, if they're all 1 millimeter inside, then the surface area is 6 millimeters and the volume is 1 millimeter. But as they grow bigger, as you see, the ratio is going to get continually smaller. So for this last one, the volume is going to be 64 and the surface area is going to be around 96 millimeters cubed, and because of that, there's going to be a huge disparity. So the main thing is the nutrients and waste. Because the cells take in nutrients from the outside, and then they have to export waste to the outside. If the volume is too big and the surface area isn't enough, they can't supplement themselves with enough nutrients, and they can't get rid of their waste properly. So in the SAT2, they're going to ask about microscopes. The two microscopes you need to know are light microscopes and electron microscopes. The first is light microscopes. These are the type of microscopes you would see on a day-to-day basis usually. So the light microscope uses light and usually uses 2 lenses to magnify. And sometimes they ask you questions on magnification. So it depends on the lenses. So if there's one lens of 10 times and the other lens is 20 times, then you multiply those numbers and get 200 times. So the advantages of light microscopes are they can study living cells. So if we look under a microscope, we can see bacteria moving if we put it in water and things like that. But the disadvantage is at high resolution. So if we magnify it 1,000 or several thousand times, it gets very blurry. Okay? And then a lot of the times we often need staining or phase contrast to see particular parts of a cell because these light microscopes, the cells are generally colorless. So we need to put some sort of contrast in, whether it be color or just shades of black and white, to show that there are different parts of the cell. So we have a picture of a basic light microscope here. They do ask you questions about the parts of a light microscope. So on the top we have the ocular lens. Here we have the objective lenses. Okay, there are generally 2 or 3 of these that you can switch around. Then we have the stage where we put the specimen and where when we look through, we can see. And then we have the condenser lenses. And then finally we have the light source. You just need to know what each particular part is. And here we have 3 different lenses where we have the specimen and then the light source. So the key parts are there are lenses and there's a light source, and that's what makes up a light microscope. The next type of microscope is the electron microscope. These can see things even smaller than cells, such as tiny proteins. Okay? Proteins are made by cells and they're only going to be a fraction of the size of actual cells. So the advantages of this electron microscope is they can observe smaller items in cells, but there are disadvantages. These cannot observe living things. So if you want to see something moving around, then you have to use the light microscope. The other disadvantage is they're extremely expensive. They're going to be somewhere on the magnitude of 1,000 to 10,000 times more expensive than your normal light microscope. So now we're going to get into the types of cells. So the first type we need to know is prokaryotes. Generally, these are less advanced, less complex, and less efficient than your animal cell or plant cell or any other eukaryotic cells. These are going to be the first cells to ever appear on Earth. Generally, you can think of them as bacteria in sulfur vents or very hot vents. So generally, all prokaryotes are bacteria. We're going to have the archaebacteria and eubacteria, which we'll learn about later on when we're learning about the various kingdoms. But other than these, there's the blue-green algae that's the exception. Okay, so down here we have a basic picture of a prokaryote. So right here, this circular thing is a DNA. Okay, this is a DNA of bacteria. So when we think about cells, generally we think, okay, they have a nucleus and inside they have their DNA. But inside a prokaryote, they're just floating around inside. There's only a ring of DNA, which is why they're much less complex. Okay? And then just generally, everything that fills in the cell is called the cytoplasm. Okay? This is a liquid inside the cell where everything floats around. Finally, these tiny things, these tiny white dots here, are called ribosomes. Okay? So these ribosomes are very important because these are what are going to create proteins. They're what are going to make the proteins for the cell to use, whether they need things inside and outside the cell. We'll get to that in a little bit, or in a later lecture. And then we have, on the inside, we have the cell membrane. So what's important about the cell membrane is this is where it keeps things inside or outside of the cell. This is very important because we can't just let anything inside and outside of a cell, or else we're going to have just a various mix of things. This is made up of a phospholipid bilayer. We'll get to that in the next lecture. And then finally we have the cell wall. Okay? So prokaryotes have a cell wall generally. These are going to add support, and it's going to be more rigid than the cell membrane, so it's going to add support to the cell. It's going to help it keep its shape. And then down here we have flagella. And these little strands here are called cilia, or pilus. Okay? Both the flagella, cilia, and pilus, they're all going to perform the same roles. They're all going to help move the cell around to where it needs to get. So, as we discussed before, in each cell of the prokaryotes, we have different organelles. It's the cell membrane. It's what protects the cell, the DNA, ribosome, cytoplasm, the flagella, and the cell wall. And generally, they're going to be made of something called peptidoglycan. It's a term that can come up, so you should remember this term. It's just what the cell wall of certain prokaryotes are made of. And what's interesting about prokaryotes is because they're simple, and less complex, they can actually survive in more extreme conditions. So maybe if it's a lot more acidic, or maybe even if it's really hot in temperature, they can survive, whereas a normal person or animal or plant couldn't survive because of their specializations. Next, we have the type of the eukaryotes. These are large and more structurally complex. Everything you can see, every living organism you can see is going to be made up of eukaryotic cells. Okay? Some of the differences are they're larger and more structurally complex, but they have more membrane-bound organelles. What this allows is that each organelle can specialize. They can perform their own duties rather than everything being mixed in together. And then this raises efficiency a lot. So if we can think about these membrane-bound organelles as parts of a human body. If we have a stomach to digest, and then we have a heart to do the heart and the brain to do other functions. But in the prokaryotic cells that we saw before, everything is just mixed in together, so there's not really any degree of specialization. So this is the primary difference that you need to remember, and what this helps with is specialization and efficiency. Okay, so if we look at the cell differences, the nucleus, we have the nucleus inside the eukaryotic cells, but we don't have a nucleus inside the prokaryotic cells. We only have a ring of DNA here versus a nucleus where it concentrates all the DNA into a specific area. Next is membrane-bound organelles. As you see here in the cytoplasm, there's just a bunch of different things floating around. Whereas here we have specific structures that do their own roles. And finally, the complexity. Just by looking at the picture, you can tell that this is a lot more complex than this. So this is the end of this section. I'll see you in the next lecture. It's going to be cells part 2, so we're going to continue on with this section. Again, I'm A1 Prep's teacher, Topcue Lee.